I am Hokuto Nakata from Hokkaido University, and I chair this seminar today. And before we proceed to the presentation from Dr. Muzandu, today's speaker, so let me introduce about his background and maybe the today's topic. So the today's speaker is Dr. Kampe Muzandu. So he's now a lecturer at the School of Veterinary Medicine at the University of Zambia. And he finished his PhD in the School of Veterinary Medicine, Hokkaido University in 2005. And currently now he's in Japan as an invited special invited lecturer at the School of Vet, Hokkaido University. And the topic for today's presentation is environmental pollution in the Lake Kariba. So the Lake Kariba is along with the border side between the two South African countries of the Zambia and Zimbabwe. And he's studying the possible contamination and its health effect on the maybe the aquatic life and their immediate environment. So maybe now you can find maybe the poster for today's presentation, and I hope you went through the abstract we circulated earlier. So okay, I think maybe that's time for my the talk. So Dr. Muzandu, so whenever you're ready, maybe you can start, please.
Dr. Nakata, thank you very much for the introduction. I appreciate it very much. And I've already been introduced, so I'll get right to it. So our study is on the chemical contaminants on the lake, and as it was said, this is a lake that borders Zambia and Zimbabwe in Southern Africa. Okay, so we want to look at the chemical contaminants and possible implications on human health as well as fish health. Okay. So the research, this particular research is a collaborative effort between UNZA and the University of, or rather the Norwegian University of Life Science in Norway. And the team members there include Jan and Steven. And from the Zambian side, mainly it's Chalumba, shown on top there, and Loziwe, and myself. So Kariba, Lake Kariba is actually a huge lake. If you're on it, you'd actually think you're at sea. It's the world's largest man-made water reservoir. It was created about 60 years ago for the sole purpose, or rather for the main purpose of then it's assumed many uses as well. So this picture just shows Zambia and lake on its southern side there, bordering Zimbabwe. Yeah, and so this is the lake, and depicting the boundary there between on the Zambian side and the Zimbabwean side on the south. Actually, Zambia has less than 50% of the lake, about 45%, and Zimbabwe 55%. So what are the uses and uses of this lake? Well, other than the hydroelectric power, there also supplies to the cities, major cities along the lake. On the Zambian side, this includes Siavonga, and on the Zimbabwe side, Kariba town. It's also used for agricultural needs, for aquaculture and its associated fisheries, for lake transportation, and also to support wildlife. But it's also bustling with tourism. Okay? There are a lot of tourist spots there, a lot of business set up there for tourism, and a few mining activities as well. Let's talk a bit more about the aquaculture on the lake. Okay, so the fisheries subsector in Zambia actually plays a very big role in its economy. So it offers an opportunity for improved nutrition, income generation, and so on and so forth. It has been growing very rapidly, actually, such that by 2016, early 2016, Zambia was the 6th largest producer in Africa. Okay, and in the years between 2009 and 2014, aquaculture production actually almost tripled. Okay, but it has slowed down somewhat now. So this fast expansion of the aquaculture sector was due to a heavy investment in commercial enterprises on the lake. Okay. So most of these enterprises are involved in the farming of the Nile tilapia, that is Oreochromis niloticus. So our research actually sampled from this species. Okay. So what are the sources of pollution on the lake? So these sources of pollution, the ones identified include the urban and industrial activities, the mining activities, agrochemicals, and oils and waste from the boats, and of course, aquaculture itself. Okay? And these pictures just show a glimpse on the different activities contributing to the pollution on the lake. So our study investigated the occurrence of selected chemical contaminants. Okay, so these are namely the heavy metals, the essential metals or non-essential metals, and then also some pharmaceutical products are found in estrogens, and also the natural estrogens, maybe from humans, from animals, and persistent organic pollutants. Though today we won't focus much on the POPs. Okay. So the metals that we assessed in the lake. Chemical contaminants, I've already mentioned where they may come from, so I'll just skip this. There have been a few studies reported in the past on the presence of metals in fish on the lake, okay, on Lake Kariba. These studies have reported on the presence of metals in other biota as well, and also sediments from the lake. So an early study was that of Berg. And they detected the presence of metals actually in sediment and in fish around the lake. And another study, an earlier study, a later study rather, by Nakayama and his teammates was carried out in 2010. At least they published their results in 2010. And they also investigated metals in Lake Kariba, but also on another lake in sediment and fish tissues, as well as water. But the scientists, Nakayama and others, they went as far as suggesting or purporting that there was an increase in the level of certain metals, in particular chromium and nickel, in the lake. So they compared their findings with those of Berg et al. And came to the conclusion that the occurrence of these metals is actually on the increase in the lake. So this is one of the stimulus we had to actually investigate further levels of these metals around fish in the lake. So sediment, the measured metal concentrations in sediment. However, our study did not measure sediment concentrations, but measured concentrations of these metals directly in the fish. So we compared concentrations of metals in wild as well as in farmed fish. So the site, where did we get the where did we do the sampling? Okay, so this is Lake Kariba. Okay, Lake Kariba. It's a large lake. It actually extends more than 200 kilometers, okay, in length, and more than about 40 kilometers in width. It's a very large lake. We sampled about 172 fish, including wild and farmed fish, and this was around June to July 2016. And the sampling locations were 5 sites, site 1, 2, and 3. Now, sites 1, 2, and 3 represented sampling from wild fish. So these were fish that were just roaming in the lake. They were not farmed fish. And also sites further up north of the lake, that is Farm 2 and Farm 1. So these were farmed fish. So we wanted to compare the concentration of metals in these. Now, what you notice that sites 1 and 2, they're like on the other end of the lake. Okay, so they're far away from where the farm is done, except for site 3, wild fish situated near the cages, near the farms. Okay, so samples processed and sent to a campus, that is Norwegian University of Life Sciences in Norway. That's where the analysis of the metals was actually carried out. This was by inductively coupled plasma mass spectrometry, or ICP-MS. Okay. So we measured essential metals. There were actually 17 metals that were measured. These include metals like magnesium, iron, and zinc, and also the non-essential metals like mercury, lead, and cadmium. So what did we find? So the concentrations of heavy metals in muscle tissues. I'm showing graphs for 3 for iron, copper, and zinc. These are actually essential metals. You can see of wild fish far away from the farms, and then you have the fish in the farms, and also you have site 3, which is the wild fish, but situated near or sampled near the fish farms. Sorry, I don't have a pointer or laser pointer here. Okay, so for iron, the highest levels of iron were at Farm 1, and these were significantly higher than in the wild fish, in particular those from sites 1 and 2. It's a similar trend with copper. The difference was even more stark here. So we have higher concentrations the farmed fish and in fish that was sampled near the farms. Okay, so you can see a trend there. For zinc, it was a similar, so not as marked difference, but a similar trend as well. And for some other metals, molybdenum, nickel, and aluminium, again, we have a similar trend for molybdenum, okay, with higher concentrations in the farmed fish, but also in the wild fish near the farms, okay. But when it came to nickel and aluminium, the scenario is a bit opposite to that of the others. So for nickel and aluminium, we found higher concentrations in the wild fish. Okay. For silver, cadmium, and lead, we see that the trend continues here with higher concentrations in the wild fish. Okay. For cadmium, see levels that are at Farm 2. Farm 2, or rather Site 2, the levels were quite high, significantly higher than the other places. This is probably due to some activities at Site 2. There's some mining activities occurring near this region. And for lead, For lead, there was a significant difference. Lead levels were significantly low, I'd say 3, significantly lower than the other places. Okay, and just of note, maybe for cadmium, is that the concentrations, in fact, the concentrations of most of these metals were not so high, such that they were below the maximum limits, that is the MLs that are set by regulatory bodies like WFOW and the EU. So these concentrations were actually low, so meaning that they do not really pose a health hazard for humans if they were to consume these fish. Okay? Findings for mercury. For mercury as well, for Site 1, we see higher concentrations of mercury in Site 1. And much higher, in fact, Site 1 and Site 3 was higher than in the farmed fish. Okay, so again, the explanation for this may be in the activity occurring there. I think Site 1, in fact, for Site 1, it's the coal mine which may be responsible for this. Mercury levels in the fish were significantly higher, so in the wild fish than in the farmed fish generally, and the highest level was 0.021. So interestingly, this figure is slightly more than the EQS standard that is set. So the implication for this is that fish in Site 1 may actually be affected adversely by this level of mercury. So in this case, this is the only probably problem with the metal concentrations in this area. So that's for Site 1. Okay, so the concentrations of metals in the mussel in wild and farmed tilapia were generally below the threshold values for adverse effects, okay, except for mercury. So the influence on capture fisheries, I mentioned that the study found significant differences in metal concentrations between farmed and wild fish. Okay, so essential metals generally were higher in the farmed fish than in wild fish. Okay, so this could be explained from the fact that essential metals are normally added to fish feed. Okay, they're normally added to the fish feed. That's why they could be higher in the fish from the farm or from the cages. Okay, so that's the explanation for this. Okay, so and we can see that in the fish in Site 3 as well, these essential metals were also higher. It could be as a result of scavenging feed from the fish farms. So feed would spill over, and these wild fish would have access to it. Okay. Okay. So that's the finding with the metals. Then the occurrence of POPs, the persistent organic pollutants in farmed and wild tilapia. So analysis of organochlorinated pesticides, PCBs, or polychlorinated biphenyls, and PBDEs, polybrominated diethyl ethers, was carried out. Again, the sample analysis was done at the Norwegian University of Life Sciences. So these were analyzed by GCMS, and then the organophosphate flame retardants were analyzed by LCMS. Okay. And a peek into the findings of that, we're going to data. The general trend is that the levels of these POPs, as measured in nanograms per gram, were lower in the farmed fish than in the wild fish. So wild fish had higher levels of POPs. Again, probably this is because of their longer life. So the farmed fish, normally by 6 months, they have to be harvested. But the wild fish, they may go on probably up to 9 months or longer. and this will give them an opportunity to accumulate these POPs. Another study we did was on the presence of estrogenic hormones in Lake Kariba. Okay, yeah. So this is an issue of emerging contaminants, and these estrogens, well, they're considered endocrine disrupting compounds. So the estrogens, they're hormones produced by mammals. They're natural and synthetic. So the natural ones include estrone, that's E1, estradiol, E2, and estriol, E3. Okay. The synthetic one basically is E2, that is 17 alpha ethanol estradiol. Okay. So the E2 basically is a component of contraceptive pharmaceutical compounds. Okay. So why study of the estrogens? So the estrogens are known as endocrine disrupting compounds, EDCs. So they may induce biochemical changes and cause loss of male characteristics in fish. Okay, so levels as low as 1 nanogram per liter can actually induce the synthesis of vitellogenin. So this is a protein found in the liver in fish. It's the reproductive protein. So sources of estrogens include domestic sewage discharge, effluent from hospitals, effluent from livestock as well, even wildlife. Okay? So the effects of estrogen on fish include induction of the vitellogenin synthesis, feminization of males, intersexuality, reproductive failure, and they may contribute to a wiping out or an extinction of an entire species of fish. Okay? I have a study from Guzel et al. It is from 2006, who showed the effects of feminization of fish due to estrogens. Okay, so they showed on top there is a male fish, and we see these fish undergoing feminization. So in this case, you have the gonads here, and these have probably 100% sperm. And in this case, you can see a reduction of the level of sperms in this male fish. So you have 50% sperm and 50% eggs in the gonads. And in this case, you have it's almost 100% eggs. Is complete feminization of the male fish. Okay. So objective of this study was to determine the occurrence of these hormones in water and sediments in the lake. So in this particular study, estrogens were not measured in the actual feed, or no biomarkers that were actually measured in the actual fish. So yeah. And another objective was to identify the possible hotspots and sources of hormone contamination in the lake. So to do this, first of all, a questionnaire was developed and administered. Okay, so these questionnaires were given to the Southern Water and Sewage Company in Siavonga. They also administered to the various lodges, or rather selected lodges along the shores of the lake, to health facilities, and to some users of contraceptives. So findings from the questionnaire. First of all, from the sewage company, it showed us that it had about 1,300 customers in Siavonga, and this was a pretty low number compared to the population of Siavonga. The population of Siavonga is about probably just over 18,000 people living there, but Siavonga is just one of the towns in the district. So the Siavonga district itself has a population of more than 50,000. So, and also the company undergoes on-site disinfection of soakaways and pit latrines, and also dislodges septic tanks that become full. Then from the lodges, there are 7 active lodges along the shores of the lake. There was compliance by these lodges in that they had soakaways and septic tanks. Others engaged the Southern Water and Sewage Company or other private companies to dislodge the waste. Then 2 other lodges admitted to actually defaulting by allowing full septic tanks to overflow, which was an unfortunate situation. So some of these septic tanks overflow, then the old pipes also would get damaged and burst. And this would lead to waste finding its way into the lake. Okay, so all these are sources of waste into the lake. The Siavonga District Hospital and the clinic, these administer oral pills, contraceptive pills, and also found that the preferred pill or contraceptive was the combined oral pill. That is, it's combined EE2 with progestin. Okay, disposal of expired contraceptives is usually by burning. So this figure shows the relative use of these contraceptives and using these, and we see that the highest number used, or most of the people used medroxyprogesterone injections, and then this was followed by the combined oral contraceptives. Okay. So this would have been the contraceptive of concern in this study, since it contains the synthetic estrogen EE2. Okay. So sample collection, the sampling took place at different sites. This is along the shores of Siavonga town. There was 3 sites there, Kanyelele, Game, and Zesco. And also the farms, some farms, there are 2 commercial farms that were sampled. And also at Lufua River, this was the nearest river that fed into the lake. And also an island at Sampa Karumba. Okay. So these are the sampling areas. And I'll show you here, Lake Kariba. And so this is Lake Kariba. And enlarging this, we have this figure. And so most of the sampling was on the southern, or rather the eastern part of the lake near Siavonga. Okay, so this is Siavonga would be around here, and other towns like Sinazongwe, major towns, Sinazongwe, Gwembe, around here. So the sampling was on this side of the lake. And so these are the sampling sites. We have a bigger region here. So these along the shores, sampling was along the shores. The farms are located here. So the cages are here. Is one farm, one large commercial fish farm with cages, and another one here with cages as well. And then you have residential areas here. And Sampakarumba is shown here as well. So those were the sampling sites. And oh yes, and another sampling site was a region we thought might serve as a control site. This is Lufua River here. So this one was, I'd estimate, about 50 kilometers away from the farms. Sample collection and water sediment. I won't go into the processes, but there was solid phase extraction of the sediment water samples. Okay, and the method that was used to determine the levels of estrogens was ELISA, as competitive ELISA was used. So this was for E1 and also for total estrogens. So what we found was that total estrogens, E1, E2, E3 combined, were detected in both the water and segment samples. But for the E1, was only detected in the water samples. The synthetic EE2 was not detected in any of the samples that were analyzed. Okay, so that's the synthetic estrogen that would be found, for example, in contraceptives. So we didn't find this, we couldn't detect this by the method that was used. Okay. Probably shed more light on these concentrations. Generally, this set of data was a little difficult to analyze because it was really unexpected, really not really expected concentrations that were found in the different sites. However, what we can say is that the concentrations that were found were below the proposed or predicted no effect concentrations. Okay, so the predicted no effect concentrations that is 6 to 60 and 0.1 nanograms per liter for E1, E2, E3, and E2 respectively. Okay, so except for the total estrogens, in some areas, in case of estradiol, the predicted no effect level is 2 nanograms per liter. But in case of the total estrogens, if you still consider this something above the 2 nanograms per liter, okay, of the total estrogens, and one, the no effect level, that is the effect at which you expect not to see any adverse effects. For E1, the level is 6 nanograms, but in this case, all these concentrations were below this level. So the presence of E1 and total estrogens points towards some contamination of the lake, though, with natural hormones. And we purported that the sources of these could be the aquatic environment, could be from humans as well as wild animals. Okay, so for instance, I take you back to the table, Lufua River. We expected to find very low levels at this point, but we could detect estrogens. And this could be because the point at which we sampled, there was a lot of wildlife. There were quite a number of hippos and also crocodiles there. So this could contribute to the levels of estrogens found there. Okay. And also another source of these estrogens in these waters could be the discharge of untreated effluent from residential areas. So it was quite an unfortunate situation that we see leakage or seepage of untreated effluent from residential areas, and this finds its way into the lake. So this is another source of estrogens. Okay. So in conclusion, the farmed fish had lower levels of pollutants compared to the capture fisheries. An exception was for metals like copper, zinc, and iron. So these were higher in the farmed fish. Then the persistent organic pollutants that were found, these could cause adverse effects. So we found that persistent organic pollutants actually were higher in the wild fish. Okay, but I would like to note also that this study is actually still ongoing. So there's still some laboratory analysis that are still ongoing. And then the estrogens are present in the lake, and we need to carry out more studies to investigate the presence of estrogens, and probably maybe with more sensitive techniques, and probably also to carry out studies of biomarkers in the fish themselves. For example, vitreogenin synthesis. Okay. So the main anthropogenic activity we determine here is due to the raw sewage discharge into the lake, also some aquatic animals, and possibly fish feed. Okay, so fish feed could contribute to the levels of some of these chemical contaminants, and especially the estrogens as well as the metals. Okay. There are a few recommendations that have tentatively come up from these studies. For example, there should be regular monitoring of pollutants in fish, in water, and in sediment as well. This is for public health safety purposes. And also, there should be regulation of the use of chemicals that are used in agriculture, in industries, and also in the fisheries sector. And proper disposal of domestic waste and industrial waste should be implemented. The disposal of the sewage system is better actually on the Zimbabwean side, where they've heavily invested in sewer ponds and septic tanks, but we need to see a similar investment on the Zambian side of Lake Kariba as well. Okay, and also to investigate the extent of contribution of fish feed to feminization of fish, if at all this is discovered, and also just to raise awareness to stakeholders, that is, the users of the lake, on the destructive impacts of hormones on fish, and also and so in the process prevent any discharge of waste into the environment. Okay. Thank you very much for listening. That's the end of my presentation.